Our today's point for discussion is recombination in bacteria. So friends, I would like to tell you that bacteria has ability to recombine its genetic material easily, and keeping in mind this property of bacteria, here there are three ways to recombine the bacterial genome, and those three ways are: first one is transformation, second one is conjugation, and third one is transduction. And we'll see one by one what is transformation is. The transformation is the process where a segment of DNA from another bacterial cell, what I have said here is DNA from donor cell, it comes in the host cell, and this foreign or donor DNA comes in contact with the host genome or double-stranded helical DNA structure, and the ultimate effect of that is the recombined, newly formed, transformed cell. So here the transformation process is completed where the foreign or donor DNA is combined with the host DNA, and this is now a new or transformed cell of bacterium. And now the second point, and that is conjugation. For understanding the process of conjugation, what is it? Conjugation is the process where DNA is transferred between bacteria through tube. Now for understanding this process, I have drawn these figures where at least two bacterial cells are required with their own genetic makeup. And here is one bacterial cell, this one is another bacterial cell. The one cell with the plasmid. Now what is plasmid is? It is extra chromosomal self-replicating small circular segment of DNA. And during the process, what happens is this plasmid can enter and recombine with the genome of the another cell. And to understand this, I have drawn this figure where the plasmid, this plasmid, and this plasmid through the tube or pill, this is the tube through which this plasmid can enter, can multiply and enter into the another cell and result in the formation of recombined bacterial cell. And here we can see the difference between these two and these two. So this is the resulted conjugation, resulted cell with the recombined form. And the third one is transduction. Now to understand the transduction process, what exactly it is? It is the process where DNA is accidentally moved from one bacterium to another by a virus. So the virus is very important and very common bacteriophage. This is what the bacteriophage virus. It is with its own genetic material and it comes in contact with the bacterial cell. Now what happens is, as they comes in contact with the bacterial cell, the DNA of this bacteriophage enters into the bacterial cell and it kills the bacterial DNA, that is chopped into pieces. So what is the original DNA of bacteria? That is broken down into the pieces, and the next stage of this viral DNA is multiplication of their own inside bacterial cell. So what happens is, few viruses pick up pieces of broken down bacterial DNA during the process, and what we can observe over here is, instead of the original bacterial genome, this genome is replaced by the entire viral DNA. And now the next stage which we can observe is, when those bacteriophages or virus infect a new cell, they give it a new piece of bacterial DNA. Now, how we can say this? Because during the process, the pieces of bacterial DNA are remnants, and this can given away along with the bacteriophages, and that segment of DNA can be transferred from that bacterial cell to another bacterial cell. And after their infection, what happens is, the segment of DNA that is carried through the viral DNA, that is bacteriophage, in the another bacterial cell, and now this becomes a transduced cell, that is the recombined DNA that enters into the next bacterial cell. And this is all about the topic recombination in bacteria. Thank you.